The chemical characteristics of fungi are widely diverse and have a major impact on local ecosystems. There are many important chemical processes that are initiated, influenced, and completed by fungi. They have evolved from unique pathways, creating a vast amount of genetic diversity in the fungal kingdom. Many of their characteristics are a result of specific chemical reactions used and experienced by each species. The culmination of these reactions are necessary for many distinct processes. The variability in their life cycle processes are due to differences in the environment, available nutrients, relationships with photobiotic partners, and whether they are biotrophic, meaning they feed on living material, saprotrophic, on dead material, or hemibiotropic, meaning they live on living tissue, and then when the organism dies, they live on the dead tissue for a while. These life cycles are here. Let's start off with the spores. Most mushrooms have haploid spores, meaning they only have half the set of chromosomes. Therefore, it's required that they mate before reproduction is, before they're able to reproduce. Their life cycle involves complex pathways of chemical processes involving spore production, the creation of a single mycelium, the creation of a secondary mycelium, and the creation of a tertiary mycelium, which can produce mushrooms. A single mycelium arises from a germ pore, which is a hole at the bottom of a spore where the first mycelium strand will grow from. A secondary mycelium arises when two single myceliums cross and mate, forming clamp connections, and they'll exchange genetic material. A tertiary mycelium arises when a secondary mycelium reproduces at a notably faster rate, culminating in the erection of a mushroom. The morphology of spores is consistent intraspecies, but may vary greatly between species. As seen here, this is the psilocybe spore, which are ellipsoid. These are the entoloma spores, irregular shaped, and the racula spores. Over here you can see a network of mycelium. The mechanisms for spore dispersal are not proven at the moment, but the most widely accepted mechanism involves what we call a sterigmata, which is this structure here. They have finger-like structures that come off. Each finger has an individual spore. A gas bubble will form, and as the pressure builds, it'll explode, and the spore will be dispersed outside the gill, and then the environment will disperse it further through wind, water, and other such. Now, fungi have the biochemical and ecological capacity to contribute to bioremediation of soil pollutants in a range of environments. The characteristics which make them suitable for this mechanism is the ability to form vast mycelia networks, the independence from using pollutants as a substrate, and their low specificity of catabolic enzymes. They have a capacity to degrade environmental organic chemicals and decrease the risk associated with metals, metalloids, and radionuclides. Those of the phyla Ascomycota, Basidiomycota, and the subphylum Mucoromycotina are noted for containing the most number of pollutant degraders. Anthropogenic chemicals, which are those which come from pharmaceuticals, agriculture, drug, food industry, and personal care products, are all pollutants from human activity, and they're all subject to fungal activity as the organic nutrients are able to be turned into a heterotrophic substrate. And the metals within these pollutants share features of micronutrients that mushrooms use. And these mushrooms are able to chemically modify these compounds to extract the nutrients that they need. And they are able to change the other compounds to increase the bioavailability for other organisms to make use of these products. And fungi are able to do this through metabolization, chemical modification, mineralization, and other transformative procedures. Fungi may also act as pathogenic, parasitic, and infectious organisms. They take advantage of unique niches and chemical processes to leach nutrients from other organisms. They excrete biomolecules for protection, ward off competition, decomposition, and other purposes. They contain what is called osteria, which is a slender projection from the hyphae, which is a single strand of mycelium, which can penetrate a tissue and therefore extract the nutrients from the host. Dermatophytes are probably the most recognizable, as they grow on skin, mucous membrane, and keratinous surfaces, and cause ringworm and other diseases. The largest known organism on the planet is a parasitic Basidiomyces, Armillaria astoriae, the honey mushroom, which is about 3.4 square miles and is estimated to be 2,400 years old. Another specimen is the cordyceps, which contains a variety of species, and they are an endoparasitoid of Ascomycota. They infect insects, and they take over the body of these insects. They colonize the insides of the body, and towards the end of the life of these insects, they somehow manipulate them to climb up higher onto plants in order to increase the success of spore dispersal. Mycorrhiza relationships have adapted to increase the availability of nutrients to plants, offer defensive mechanisms, possess the ability to chemically alter structures of proteins and nutrients to transport from one plant to another through mycelial networks. They produce secondary metabolites to act as antifungal agents, antibacterial agents, and to ward off other plant competitors. There are also many unobserved secondary metabolites due to these genes being silenced and unexpressed. There are also what are called arbuscular mycorrhizae, which are an endomycorrhizae, and they live on the inside of the cortical cells of plant roots. They respond to signaling molecules by the plant host, which are strigolatones, a plant hormone released by the plant roots upon phosphorus deficiency to induce hyphal branching and to connect with the arbuscular mycorrhizae. These signals also stimulate the production of fungi symbiotic signals, which may signal plant roots to grow, as well as other reactions. Mycelial networks are also capable of transferring nutrients from one plant to another. This is speculated to be influenced by the nutrient concentration between connected individuals and the shading level of receiver plants. So a plant that's receiving more light is speculated to be giving some of its nutrients to one that isn't. These complex relationships are vital to the success and survival of interconnected organisms. Fungi are the final piece to the cycle of life, as they are the only organisms capable of breaking down cellulose and lignin, which are prolific cellular products in our world from plants. They're able to create more bioavailable products for other organisms. The chemical properties of fungi possess an array of applicable discoveries in science. These applications have relations within medicine and apply to current global issues. Further research within this study may continue to unravel the mysteries these organisms have obtained through millions of years of evolution. Thank you. Thank you very much. And for the after show. What? I don't get it.
What?